Hello. In this presentation, I will be discussing the research entitled Spatially Resolved Characterization of Water and Ion Corporation in Bacillus spores, which was carried out at the Lawrence Livermore National Laboratory in Livermore, California. I will begin this presentation by discussing a quick introduction to the research performed, and then we will quickly begin discussing the permeability experiments, including the methods used and the results, as well as the experiments regarding uptake, exchange, and distribution of water in dormant Bacillus spores, metal uptake and distribution in the spores, anion uptake and distribution in the spores, and their respective methods used, results, and relevant conclusions. Historically, spores were assumed to be impermeable. Fortunately, recent pioneering studies have demonstrated that dormant spores are permeable to water. These studies also demonstrated that the permeability to various solutes is controlled by their molecular weight, charge, and lipophilicity. Research regarding the structural limits on permeability, particularly into the spore core, still remains an active area of interest. Water uptake properties of Bacillus spores have been examined by various laboratories, which has demonstrated that the entire spore volume, including the core, is permeable to water. However, in these studies, uptake of water into the spore was not observed directly, but rather inferred from measurements of indirect phenomena, such as change in the size of the spore after water adsorption. Imaging mass spectrometry with a nanosims, a high-resolution secondary ion mass spectrometer was used to directly probe the spatial distribution of permeating water vapor and ions in individual Bacillus thuringiensis subspecies israelensis spores following exposure and dehydration treatments. These results confirm that the entire spore volume, including the core of the Bacillus species spore previously mentioned, is permeable to water and hydrated ions. So, before I begin discussing the methods used throughout the permeability experiments, I think it's important to note how the spores were actually treated prior to experimentation. So, these spores used throughout the experiments were obtained from the Bacillus Genetic Stock Center. Prior to experimentation, Bacillus vegetative cells were prepared, purified, applied to sporulation medium, and incubated at 37 degrees Celsius until sporulation was achieved. The spores were then harvested in double distilled water, washed 6 times by centrifugation, and resuspended in double distilled water. These treated spore samples were then examined through nanosims analysis to determine the spore integrity before experimentation began. The first experiments I will be discussing are the permeability experiments. For water and ion uptake experiments, the Bacillus spores were exposed for predetermined intervals to deuterated water, otherwise known as D2O, lithium fluoride solution in deionized D2O, or cesium chloride solution in deionized D2O. The solution concentrations were measured by inductively coupled plasma mass spectrometry, or ICP-MS for short, and intact spores were mapped by scanning electron microscopy, SEM for short, for nanosims analysis. Because the spore exosporium, coat, and cortex could not clearly be distinguished from one another in the nanosims images, these structures were referred to collectively as the outer structures and will be referred to as the outer structures throughout this presentation. So these are the results from the first permeability experiments. An imaging depth profile method was used to characterize elemental distributions within individual spores. This first figure represents the distribution of carbon, fluorine, phosphorus, and chlorine in a single Bacillus endospore, as determined by NanoSIM's imaging depth profile analysis using a cesium-133 plus primary ion beam. In Figure 1a, the ion images obtained from serial scans of the spore by the cesium beam represent the summed ion counts for each species. Lighter colors represent higher counts. In Figure 1b, the depth profile data are extracted from serial images of the spore as it is eroded away by the cesium beam. As you can see on the graph, depth in nanometers is on the x-axis, while relative counts of their respective ions are on the y-axis. The circles represent carbon-12, the diamonds represent fluorine-19, the squares represent phosphorus-31, and the triangles represent chlorine-35. The data presented here are average results of multiple measurements of individual spores within a given population. We will now shift our attention to the experiments involving the uptake, exchange, and distribution of water in the dormant Bacillus spores. Water uptake was studied by exposing dry spores to saturated D2O vapor. The use of deuterium-labeled water enabled the permeating water to be distinguished from intrinsic water associated with the spore. One set of samples was exposed to D2O for times ranging from 1 minute to 15 days in sealed glass containers containing 10 microliters of D2O. Another sample was placed in a par bomb with 10 microliters at 80 degrees Celsius for 2 days to simulate longer exposure conditions. The samples were analyzed on the day of harvesting and after 3 months of residence in a vacuum chamber. Here, the spatial distribution of deuterium incorporation in individual Bacillus spores in response to the D2O vapor exposure was directly imaged, and it can be seen here in Figure 2. Figure 2a shows the distribution of deuterium and hydrogen as a function of depth in a Bacillus spore. Deuterium was found throughout the spore, which provides direct evidence of the incorporation of deuterium associated with D2O vapor into the spore core. The observed distribution of deuterium in the spore was postulated to be the result of D2O permeation and exchange within the spore volume, including the core. Figure 2b shows the summed hydrogen minus and deuterium minus images of a single spore. Similar to the images in Figure 1 shown earlier, lighter colors represent higher counts of the ions in the spore. Additionally, the level of deuterium incorporation with increasing D2O exposure time was examined. As shown in Figure 2c, the abundance of deuterium in the spore increased with increasing D2O exposure time. Analysis of the samples after 3 months in vacuum showed that the deuterium to hydrogen ratio decreased across the full range of D2O exposures, which was likely caused by the progressive loss of weakly bound D2O via dehydration or exchange with vacuum chamber H2O. So in other words, the H2O in the vacuum chamber was kind of switching places with the D2O that was previously bound to the spore. Nonetheless, these results supported the conclusions that longer time exposures led to higher levels of exchange. We will now discuss the experiments involving the metal uptake and distribution in the Bacillus spores. Cation and anion uptake was studied by exposing spores to a lithium fluoride or cesium chloride solution in D2O for exposure times ranging from 5 minutes to 4 days. The choice of cations was based on the observation that both lithium and cesium have low abundances in native spores and also provide a range of ionic radii. Following exposure to either a lithium fluoride or cesium chloride solution, spores were washed several times in deionized water to remove residual solute. Prior to NanoSIMS analysis, the samples were imaged with an optical microscope and/or SEM to identify and map regions for analysis. The permeability of the spores to water vapor suggests that hydration pathways or channels may provide a mechanism for diffusion of ions within spores due to the increased mobility of hydrated ions. This permeability was examined by using hydrated monovalent cations, lithium plus and cesium plus, in deionized water. As stated earlier, both lithium and cesium have low abundances in native spores. Therefore, their uptake by spores should be readily identifiable. Exposure to a 10 micromolar lithium fluoride solution resulted in permeation of lithium throughout the spore, showing that the spore was permeable not only to water, but also to small ions such as lithium plus in solution. Following 3 days of exposure to a 10 micromolar lithium fluoride solution, the lithium content of the spores increased by a factor of roughly 10 orders of magnitude compared to that of the unexposed spore, which can be seen in figure 3a. NanoSIMS lithium depth profiles of exposed spores showed that lithium concentrations were higher in the spore outer structures relative to the core. Figure 3b shows the NanoSIMS ion images of spores with no exposure, while 3c shows the NanoSIMS ion images of spores with 3 days of exposure, both to 10 micromolar lithium fluoride solution. The calcium images show the locations of the spores, and the lithium images show the low background in the control sample shown in 3b, and that lithium is localized in the spores in the exposed samples, which is seen in 3c. In a second set of experiments, lithium uptake in hydrated spores as a function of exposure time was examined, and it was found that lithium content increased steadily with increasing exposure times extending into days, which can be seen in figure 4a. This is indicated by the increase in the measured lithium plus to carbon plus ion ratio over time. Additionally, the average lithium distribution profiles in spores for the experimental exposure times are shown in 4b. In all cases, lithium content was higher in the spore outer structures than in the core. Similar permeability experiments were performed with spores suspended in cesium chloride solution. Unfortunately, unlike lithium fluoride, exposure to the cesium chloride solution resulted in a significant portion of the spores to become phase dark. SEM analysis showed that these spores were observed to have lost a large portion of their contents and had deflated. Intact spores were examined, and depth profiles showed that cesium chloride exposure led to permeation of cesium plus throughout the spore, with more cesium accumulating in the spore outer structures. As with lithium fluoride exposure, the cesium content of intact spores increased with continued exposure over a period of several days. However, the temporal behavior of cesium uptake in spores was not pursued in detail due to the lysis effect of exposure to cesium chloride. So we're going to now shift into a discussion based on the results that were just presented in these experiments. So the results presented here demonstrate that water and monatomic ions in solution permeate Bacillus spores into the core through the exosporium, coat, and cortex. Upon exposure to aqueous solutions of lithium fluoride and cesium chloride, measurable amounts of cations and anions are retained by the spore, resulting in time-dependent net ion uptake. It was postulated that the uptake of solvated ions is the combination of two equilibrium processes, which include movement of ions into the spore in response to a chemical gradient or hydration gradient, and also adsorption at available sites within the spore via an exchange mechanism. So for cations, which I have here, the equation x plus YR is in equilibrium with Rx plus Y plus applies, which I have written out on this slide. So X plus is the permeating cation, R represents an organic ligand at the spore exchange site, and Y plus is another cation or the hydronium ion. With dehydration, the majority of unbound ions in water are presumed to leave the spores, in which it is postulated that charge balance is maintained by the co-propagation of cations and anions. I'm going to go back to the data. So, in the hydrated spore experiments, the lithium uptake profiles showed gradients, which can be seen in figure 3b. But the gradient structure was independent of exposure time, and lithium content increased with increasing exposure time. On the basis of these observations, it was concluded that under hypotonic conditions, uptake of cations in Bacillus thuringiensis subspecies israelensis spores is limited by the rate of exchange. Under these conditions, elemental abundances should increase as a function of exposure until all of the exchange sites are saturated. These results were not surprising, since previous studies on bacterial spores have led to similar conclusions. These results confirm that diffusion is relatively rapid, and solvated cations permeate the entire spore, including the core, on a timescale of seconds to minutes, in which 5 minutes was the shortest time point studied. The observation cation gradients in spores indicate higher concentrations of monovalent cation exchange sites in the outer structures relative to the core. Bacterial cell walls are known to absorb cations. They contain carboxylate, phosphate, hydroxylate, and amino-functional groups that serve as exchange sites for metal cations via deprotonation. Carboxylate and phosphate groups are negatively charged under hydrated conditions, which allow them to be potent scavengers of cations. In Bacillus spores, the spore structure is characterized by several proteinaceous spore coat layers and by cortex and core wall peptinoglycan enclosing the central core region. For species such as Bacillus thuringiensis subspecies israelensis, the coat is covered by an additional outer layer called the exosporium, which is primarily composed of proteins. All of these layers contain potential exchange sites for cation uptake. Potential exchange sites for cations in the core are phosphate and amino groups that are associated with the spore DNA, small acid-soluble spore proteins, and dipicolinic acid, otherwise known as DPA. Based on the high abundance of DPA in the spore core and the relatively low retention of lithium and cesium in the spore core seen in these experiments, spore core DPA does not readily bind monovalent cations under the conditions used in this study. Okay, so we will now begin talking about the anion uptake and distribution in the spores. So, in conjunction with the lithium and cesium uptake studies, the uptake and distribution of the associated counter ions, fluorine minus and chlorine minus, was examined. These studies were affected by several factors, which I will talk about now. Unlike the extremely low abundances of lithium and cesium in native Bacillus spores, fluorine and chlorine abundances are significant, typically with higher abundances of these species in the spore outer structures relative to the core. Excuse me. Therefore, both fluorine and chlorine were detected in the spores prior to experimentation. In addition, chlorine is a germinating agent and therefore was potentially responsible for the observed lysis of a significant portion of the spore population in response to cesium chloride exposure. So, I'm now going to discuss the graph here. So, figure 5 shows the average fluorine content in the spore core and outer structures following lithium fluoride and deionized water exposures. 3 concentrations of lithium fluoride were used, which included 0.4, 10, and 40 micromolar. Both dry and hydrated spore preparations were utilized as well. Prior to exposure, spores had higher fluorine content in the outer structures relative to the core. Figure 5b shows exposure to 0.4 and 10 micromolar lithium fluoride solutions, which resulted in the loss of fluorine from the spore outer structures, while the abundance of fluorine in the core remained relatively unchanged. This led to a reversal in the spore fluorine distribution pattern, with the core having a higher fluorine content relative to the outer structures. In the case of 0.4 micromolar lithium fluoride exposure, the spore fluorine content remained relatively unchanged over time following the initial fluorine loss from the outer structures. Exposure to the 40 micromolar lithium fluoride solution resulted in a significant increase in the core fluorine concentration. Figure 5c shows that exposure of dry spores to deionized water led to an initial removal of fluorine from the spore as a whole, following which the overall fluorine content and distribution within the spore remained unchanged. Figure 5a shows the average fluorine content measured as a fluorine to chlorine ratio in the spore outer structures and core as a function of exposure time to 0.4 micromolar lithium fluoride solution. You can see that exposure time is exhibited on the x-axis, while the fluorine to chlorine ratio is exhibited on the y-axis. In all of the lithium fluoride treatments, the spores developed higher fluorine abundances in the core relative to the outer structures, and I am back to referencing figure 5b. This reverses the initial fluorine distribution observed prior to the exposures. This was interpreted as an equilibrium state of the spore relative to the surrounding solution. Figure 5a and c show the time-resolved data that was collected for the 0.4 micromolar lithium fluoride and deionized water treatments. These data show that changes in spore fluorine abundance occurred rapidly relative to changes in the spore lithium concentration, which was shown earlier in figure 4. However, the fluorine data are not directly comparable to the lithium uptake results. Figure 6, which is shown here, shows the chlorine data obtained from the lithium fluoride and deionized water exposures, which showed permeation of chlorine into the spore. However, while the spore chlorine content was altered by these exposures, the overall distribution of chlorine remained unchanged, and with the outer structures having higher chlorine content relative to the core. Figure 6a shows that the chlorine contents of both the outer structures and the core were reduced in the 0.4 micromolar lithium fluoride treatments, which was similar to the starting value. Figures 6b and c show that the 40 micromolar lithium fluoride, 10 micromolar lithium fluoride, and deionized water treatments, the chlorine content unexpectedly increased. A potential explanation for this is regarding the starting spore material. Treatments involving dry spores resulted in increased spore chlorine content, while those with hydrated spores did not. It was concluded that whatever the source of chlorine in these treatments, the data demonstrate that the chlorine contents of both the spore core and the outer structures can be modified relative to the starting composition, and that the outer structures maintain higher chlorine content relative to the core. So I will now go into a discussion part of the results just presented. This data indicates that anion incorporation is also controlled by equilibrium binding, which can be seen in both figure 5 and 6. So this is figure 5, this is figure 6. Potential anion binding sites include proteins and cations. The difference in fluorine minus and chlorine minus equilibrium distributions may be explained by the affinity of F minus for calcium 2+, which is abundant in the spore core. The difference in the distribution of cations and anions in the spores demonstrates that elemental incorporation is independent of the counterion, even if copropagation maintains charge balance. The anion data in figures 5 and 6 also indicates post-production accumulation of contaminants in the spore outer structures, which can be removed with washing. The presence of these contaminants is relevant because the starting samples were washed repeatedly in double distilled water and stored in double distilled water in the case of the hydrated spores. This observation indicates that accumulation of contaminants could be used to reconstruct the post-production history of a sample. More work would be necessary to understand the potential sources and the exact information that could be gained. Under standard growth conditions, calcium is the most abundant metal in Bacillus spores because it is chelated by DPA in the spore core. Previous work has shown that the abundance of calcium, other elements will substitute for calcium, demonstrating that the elemental composition of bacterial spores is strongly influenced by the elemental composition of the media used to produce the spores. It has also been shown that calcium can be removed from spores by acid treatments and replaced by other elements. And so this study, and this is more of an overall discussion of all of the research that was done. It demonstrates that elemental incorporation can occur after spore formation through solute permeation without special treatment, and therefore must be considered when analyzing the elemental composition of bacterial spore samples. The monovalent cations are preferentially incorporated in the outer structures of spores, but also penetrate to and accumulate in the core. This data demonstrates that permeation is rapid relative to incorporation, and the primary control of the distribution of permeating elements in a spore is the availability of binding or exchange sites. Because the rates of incorporation found here are low relative to standard processes such as washes, the level of incorporation potentially could be used to determine the time of exposure under known conditions. And finally, these results demonstrate that elemental abundances and distributions in spores are influenced by the chemistry of the local environment, and therefore present novel opportunities for environmental microbiology and microbial forensics. These results also demonstrate the need to collect samples and prepare them for analysis using methods that minimize the alteration of the elemental or isotopic signatures of interest. So this was the paper, and I want to thank you for watching, and that is all from the paper that I had to sift through in order to complete some of my senior research.